My great pleasure to introduce Ghida Baalbaki, who is a PhD candidate in biology, also at INRS, so the National Institute of Scientific Research, if we anglicize it in Quebec. She holds a bachelor's degree in life and earth sciences, biochemistry from Saint Joseph University of Beirut, and a master's degree in reproductive and developmental biology from the University of Paris, Université de Paris. Her doctoral research is conducted under the co-supervision of Professors Géraldine Delbes, Cathy Vaillancourt, and Élise Caron-Baudouin, and it focuses on the effects of contaminants associated with UNG exploitation on pregnancy and fetal development. Alongside her research work, she has taken part in science communication and knowledge transfer activities to raise awareness of EDCs and environmental health. So as a consequence for her excellent work, she was awarded this edition's CIAPE-EVERTEA Doctoral Prize. And we look forward to learning more about her work in a talk entitled Trace Elements Associated with Unconventional Natural Gas Disrupt Fetal Steroidogenesis, Evidence from In Vitro Models. And I won't forget to stop sharing this time.
Thank you, Jan. I will share my screen.
Excellent.
Do you see the full screen? Okay, perfect.
Yeah, we see the screen.
Okay, perfect. Thank you.
It's not the full, sorry, Ghida, it's not the full one yet. So yeah, now it's good. Okay, perfect.
Oh, okay. So thank you, Jan, for the introduction. So I'm Ghida. I'm glad to present you today my work about the effect of trace elements associated with gas exploitation on fetal steroidogenesis using in vitro models. Okay. So the northeastern British Columbia, and more precisely the Peace River Valley, is an area of intense and conventional natural gas exploitation by hydraulic fracturing. This process consists on injecting large volumes of water, sand, and chemicals in rock formation to create fractures, freeing natural gas trapped in the rock. During the process, two types of wastewater are generated: the flowback water that returns to the surface directly after fracturing, and the produced water that emerges over a longer period of time. However, several studies have reported surface and groundwater contamination by trace elements near fracking sites. These trace elements could contaminate drinking water by accidental spills, wastewater leaks, or improper wastewater disposal, raising concerns about their impact on human health. In 2019, a pilot study done by our team detected high concentrations of 5 trace elements: aluminium, barium, cobalt, manganese, and strontium, that were between 2 and 16 times higher in urine and hair samples of pregnant women residing in the northeastern British Columbia. This study was the catalyst of a bigger project, the EXPERIVA study, or exposure in the Peace River Valley, which is a biomonitoring study that aims to assess the exposure to trace elements in a bigger cohort of 85 pregnant women residing in the northeastern British Columbia. In 2023, it has been reported that a significant proportion of participants exceeded the 95th percentile of the reference population, 58% for barium, 73% for cobalt, 28% for manganese, and 60% for strontium. So trace elements are present naturally in the environment and our food, but at very low concentrations. Some are essential for human health. However, excessive exposure could lead to adverse health effects. For example, manganese is an essential element for metabolism. However, it was associated with neurological defects in children. Cobalt is also important for red blood cell production, but it was classified as environmentally toxic by the Environmental Protection Agency. Other trace elements are non-essential, like aluminium, barium, and strontium, and excessive exposure could harm the central nervous system, induce cardiovascular diseases, or impacts bone formation. Despite the evidence that trace elements can reach and cross the placental barrier, there is still a gap in the literature about the effect of in utero exposure to trace elements on fetal development. During pregnancy, steroidogenesis or steroid hormone production by the fetal placental unit is essential for maintaining a healthy pregnancy. However, studies have reported that the contaminants associated with UNG are suspected to be endocrine disruptors. So, as you know, an endocrine disruptor is a substance or mixture that alters endocrine system functions like steroid hormone production, bioconversion, elimination, transport, binding, or action, leading to adverse effects in an intact organism, its offspring, or subpopulations. Several studies have reported agonist and antagonist effects of surface and groundwater from UNG sites on estrogen, androgen, progesterone, thyroid, and glucocorticoid receptors. Pregnancy is a critical window of vulnerability for maternal and fetal health as it's a period of dynamic development and tissue modeling. During pregnancy, the placenta plays important roles in fetal implantation as protective barrier for maternal-fetal exchange of nutrients and gases, waste elimination, and most importantly for hormone synthesis and secretion, which are crucial for maintaining a healthy pregnancy. However, the placenta is highly sensitive to EDCs due to the abundance of steroid hormone receptors. Several EDCs like phthalates, PCBs, and pesticides were linked to growth restriction, low birth weight, preterm birth, preeclampsia, miscarriages, and gonadal development disruption. Studies suggest links between UNG well density and proximity and adverse pregnancy outcomes and negative impacts on fetal health like preterm birth, miscarriages, or fetal malformation. Also, studies suggest potential toxicity of UNG-related contaminants on reproduction and fetal development. However, there is still a huge gap in the literature about the specific effect of trace elements associated with UNG and their EDC potential on pregnancy and fetal development. So we hypothesize that trace elements at doses similar to those measured in Xperiva participants, alone and in mixtures, act as disruptors of steroidogenesis, male gonad development, and fetal placental hormonal communications. So our objective was to investigate the toxicity and EDC potential of trace elements associated with UNG at human fetal exposure levels on fetal development. Our specific objectives are first to determine human fetal exposure levels, then to assess the impact of trace elements on steroidogenesis and fetal testicular development, and finally to evaluate the effect on fetal placental hormonal communication and aromatase activity. So first, we have selected trace elements for which a significantly higher proportion than 5% of pregnant women in the Xperiva cohort had concentrations in urine and or hair above the 95th percentile of the Canadian reference population. So we selected 5 trace elements to test: the manganese, barium, cobalt, strontium, and aluminium. We then had to determine the fetal exposure levels to those 5 elements. So we have done a literature review of the concentrations of these elements in the umbilical cord blood from previous biomonitoring studies of pregnant women from general population. So you can see here each bar represents the range of concentrations in umbilical cord blood from one study. And it's the same for each trace element, 8 trace elements. We then did an extrapolation of Xperiva data, and we selected the experimental concentrations within and above the ranges determined from the literature. So three experimental concentrations were selected per element: 0.05, 0.5, and 5 microgram per liter for cobalt, 550 and 500 microgram per liter for manganese, barium, strontium, and aluminium. We also tested mixtures of the 5 elements, the mix 1 at the lowest concentration of each, and the mix 2 at the mid-range concentration of each. Our second objective was to assess the impact of trace element exposure on steroidogenesis and fetal testicular development. So we have used 3 in vitro cell and tissue-based assays relevant to EDCs and development. First, the transactivation assays of human estrogen and androgen receptors, ESR1 and AR. Second, the organotypic culture of rat fetal testis. And third, the human adrenal carcinoma cell line culture. So for the transactivation assays, these assays were recommended by the OECD to test EDCs, as it helps to identify potential EDCs and elucidate their mechanisms of action. These assays are based on mammalian cell line expressing human estrogen receptor, ESI1, and androgen receptor, AR. Using these assays, we can study the agonist or antagonist effect of our trace elements on ESR1 and AR by activating or inhibiting the transcription of a reporter gene, which is the luciferase. So in the first case of activation, we'll see an intense luciferase activity signal measured. And in the second case, we'll know or see a low luciferase activity signal. So here are our results. And as you can see here, the Y axis represent the percentage of receptor activity, and the X axis represent the trace elements that we have tested or the controls. In the agonist assays, the red line represents the activation threshold, which was set on 10%. And if we see a receptor activity that is higher than 10%, we'll consider it as agonist effect. On the other hand, for the antagonist assays, the red line represents the inhibition threshold, which is an 80%. So if the receptor activity is under 80%, we consider it as antagonist effect. However, as you can see here, none of our 5 trace elements nor their mixture had an agonist or antagonist effect on ESR1 or AR. We then used the organotypic culture of rat fetal testis. A quick recap of the fetal testis structure, as you can see here, it's mainly formed by seminiferous cords that you can see in white and contains gonocytes or germ cells in yellow, surrounded by Sertoli cells. In the interstitium, we can find the Leydig cells that are responsible of testosterone production, and which is crucial for maintaining a healthy masculinization and fertility. So the organ culture of rat fetal testis was widely used to assess the toxicity of various EDCs on fetal testicular development, gametogenesis, and steroidogenesis in terms of age, time, and dose. So our model was the Sprague Dolly rat. We've collected the fetal testis at 15 days post-conception, which means in the masculinization programming window and the window of sensitivity to xenoestrogens. We then put the fetal testis on inserts floating in culture media containing or not the trace element to test. The culture was maintained for 3 days, and the culture media was changed every day and kept for testosterone secretion dosing by ELISA. At the end of the 3rd day, we've added a lash, a stimulating hormone, for 3 hours, and we've also kept the media for testosterone secretion dosing by ELISA. At the end of the culture, we have collected the testis and fixed it for cellular analysis by immunofluorescence staining. So our ELISA results have shown that after 72 hours of exposure and 3 hours of a large stimulation, aluminium and manganese had no significant effect on testosterone secretion in comparison to the control that you can see here in white. In contrary to the MEHP, which is a testosterone production inhibitor and was used as positive control. However, strontium, barium, and cobalt increased testosterone secretion significantly at their highest concentration from 48 hours for barium and cobalt and from 24 hours for strontium. We then wanted to see the effect of these elements in mixtures. So we tested the mix 1 at the lowest concentration of each and the mix 2 at the mid-range concentration of each. And the big surprise was that after 48 hours of exposure, mix 2 increased significantly testosterone secretion at mid-range concentrations, which means at concentrations at which none of the 5 trace elements had a significant effect individually, as you can see here under the red arrow. We then wanted to see if this mixture effect was due to the 3 active elements that induced significant effects individually at high concentration, which are barium, strontium, and cobalt. So we have tested the mixture of mix 3, so the mixture of the 3 elements, but at the same mid-range concentration of mix 2. However, in contrary to mix 2, mix 3 had no effect on testosterone secretion. These results led us to suggest an interaction between the 5 elements leading to this mixture effect and possibly implicating synergistic potentiation mechanisms. We then wanted to see the effect of trace element exposures on testis structures. So we have done triple staining by immunohistochemistry for Sertoli cells in red, germ cells in green, Leydig cells in purple, and on the right you can see the merge of the 3 channels with the DAPI. So our results have shown that cobalt altered testicular structure. As you can see here on the left, it reduced the proportion of seminifer scores significantly in comparison to the control. This effect of cobalt was not only observed on the highest concentration, but also at the mid-range concentration. On the other hand, no effect was observed on germ and Sertoli cell density for any of our tested elements or their mixture. We then looked on the effect on Leydig cell density and surface. And as you can see here on the left, cobalt and strontium induced hyperplasia and or hypertrophy of Leydig cells in comparison to the control, which could explain in part the previous results that we have seen in ELISA for the increase of testosterone secretion. We finally looked on the effect of 3-beta-HSD enzyme expression. And as you can see, in contrary to the individual elements, the mix 2 of the 5 elements increased significantly 3-beta-HSD protein level. Here, I want to remind you that the 3-beta-HSD is a steroidogenic enzyme that is expressed in Leydig cells and is implicated in testosterone production, which also could explain the previous results on testosterone secretion. We finally used we tested the effect of trace elements on steroidogenesis using another model of steroidogenesis, which is the human adrenal carcinoma cell line culture, which is widely used to assess potential effects of EDCs and it has the ability to produce a variety of steroid hormones, including testosterone. So after 24 hours of cell adhesion, we've added the culture media containing the trace element to test, and we've treated the cells for 48 hours. At the end of the culture, we've collected the cells for viability test by Chipon Blue and the media for testosterone dosage by ELISA. So in this essay, we have tested the cobalt and mixtures that induce the most effects in organotypic culture. And here you can see the results of our viability test. And as you can see, none of our tested conditions had a cytotoxic effect on H295R cells. On the right, you can see the results of ELISA on testosterone secretion. And as you can see, in contrary to what we have seen in organotypic culture, neither cobalt nor mixtures had a significant effect on testosterone secretion in comparison to the control, in contrary to the foxcholine and prochlorase that stimulates and inhibits testosterone production respectively, and was used as positive controls. So these results were the fruit of our first published article that was recently published in the Environmental Pollution Journal. So if you're interested to look deeply in the results, you can find it online. Finally, I will not go deep in the details as I don't have enough time, but our last objective was to evaluate the effect of trace element exposure on fetal placental hormonal communication and aromatase activity. So for this objective, we used the fetal-placental co-culture model that was developed by Cathy Vaillancourt team. And using this co-culture, we'll use the two cell lines, the H295R cells that represents the fetal unit and the BO cells that are choriocarcinoma cells and represent the placental unit. This co-culture has the capacity to mimic the fetal-placental hormonal communication as it can produce all pregnancy key hormones, have all the enzymatic capacities of the fetal placental unit, and has high basal aromatase activity. So, in conclusion, our objective was to assess the endocrine disrupting potential of trace elements associated with UNG on fetal development. So after we've selected the fetal exposure levels and the experimental concentrations, we have shown that none of our 5 elements or their mixture had effect on ESR1 or AR. However, cobalt, barium, and strontium increased testosterone secretion at their highest concentration. And more interestingly, the mixture of the 5 elements increased testosterone secretion at concentrations at which none of the 5 elements had a significant effect individually, and most importantly, at concentrations that are representative of human fetal exposure levels. These effects were associated with an increase of Leydig cell density and surface, as well as 3-beta HSD expression. We think that these results these effects are specific to the fetal testis, as no effect was observed in the H295R Steroidogenesis assay. So finally, the study responds to the urgent need to assess the risk associated with UNG, in particular trace element contamination, likely to act as EDCs. It helps not only to elucidate the mechanisms involved, but also to determine whether current regulations adequately protect populations, particularly the most vulnerable, including pregnant women and their fetuses. At the end, I want to thank my PhD director and co-directors, Géraldine Delbès, Cathy Vaillancourt, and Elise Caron-Baudouin, and our collaborator on the project, Marc-André Werner, my lab colleagues, our project funders, and a special thank to the CIAPE for this amazing opportunity. And thank you for your attention.
Excellent. Thank you very much, Ghida, for an excellent talk. And the floor is once again open for questions. Valérie.
Well, thanks. Yeah, maybe. Okay, thank you. Well, thank you for the very nice presentation and congrats on the award. This is always very interesting to see how metals affect, right, the endocrine system. But so which of the enzymes that are involved in all the mechanistics of interest to you are actually metallo enzyme that could be the culprit, if you want, the reasons of those activation, right? Because at this point, we know that many enzymes need metals to actually do their activity. So my understanding would be that these new metals or trace element would be actually in competition for those sites and would stimulate, because what I said, it's like you have increase in testosterone, for example. So is that something that you're considering?
It's a great question. Actually, we like if I can explain, like we didn't really look on the different enzymes, but as we have shown that trace elements that first that EDCs could act on via many pathways, not only receptors or enzymes. For example, in our case, we have shown that trace elements could increase 3-beta-HSD enzyme expression, which is implicated in steroidogenesis. But also, we have shown that it could be these effects could be induced, could be dependent of receptors on enzymes and induced by Leydig cells proliferation. We didn't look on the proliferation of Leydig cells, but we have shown that we have an increase in Leydig cell density and surface, which could be explained by proliferation, and as well as 3-beta-HSD. I'm not sure if I really answered your question, but
well, but you know, with all the positive and negative feedbacks of the endocrine system, if you have an increase in testosterone, let's say your metals expulsion would increase your enzyme activity, which would increase your hormonal, let's say testosterone, which will increase then the proliferation of your cells, which will increase the hormone, right? It's all like it's a loop. It's a mechanism of action, but it's all like dependent one on the other. So anyway, perhaps I guess it's more comments that I encourage you to look into which of those enzymes are actually metallo-dependent, and I would guess you can tell me in a few years if that was one of the reasons of this increase. Thank you so much.
Thank you.
Thank you, Valerie. And we have time for one more question, and Vicky has raised her hand.
Okay, sure. Yeah, I was going to just make a comment and ask about the literature review. So in the ecotox world, really, there's only been one metal that's been shown to have what we think is direct estrogenic activity, and that's cadmium, and induces vitellogenin in fish. And so, yeah, I guess I was curious, the literature in the mammalian world, if, I'm kind of surprised that this hasn't been tested, but obviously it's a different area. With fish, they're exposed to lots of metals downstream of metal mines. So we've looked at that. So I was curious if there was any literature supporting any of these hormonal actions or EDC actions of metals. And then I wondered if you could just explain, you mentioned the current regulations for UNG. Are you just referring to drinking water guidelines, or are there specific guidelines associated with that field that limit the amount of trace elements that can come back up?
Okay, thank you. For your first question, actually, to our knowledge, for example, for the transactivation assays, we know that there is no other study that looked on trace elements effects on agonist or antagonist effect on ESR1 or AR. Only one study that has looked on heavy metals and have shown that biome could have an agonist effect to ESR1, that it's contrary to what we have shown. But we think that this discrepancy in results could be due to the differences in models, the cell lines that we have used, and the concentration and time of exposure. Actually, in this study, they used the MCF7 cells, the breast cancer cells. So we think that the difference in the cell lines, and in our study, we've used the non-human mammalian cell line. So we think that it could be the difference in cell lines used, so the environment and the expression profiles of these cell lines are different, which could lead to different effects. So to our knowledge, this is the only one for the effect on ESR1 and AR. For other effects, I think we are the first to have shown the effect on fetal testis. And yeah.
Okay. Do we have time to talk about the regulations yet, or do we need to move on?
If it's quick, maybe
Again,
1 minute. 1 minute.
Are there special regulations for UNG for drinking water, or?
Actually, I think it will not be too long, because I don't really have deep knowledge this specific matter, but to my knowledge, that there's no regulations in drinking water for fracking, especially associated with fracking, and there is no regulations of the intensity of UNG in British Columbia, for example, or Alberta.
Thank you.
Okay, thank you very much. Congratulations again on the award and excellent presentation.
Thank you.
We're